Cellular respiration. General equation. You start out with C6H12O6, and that is glucose. It could also be fructose or galactose. They all have the exact same chemical formula. And you add oxygen, because what we're going to be doing is oxidizing the carbon in this glucose molecule. And generally, when you oxidize things, that is going to be an exergonic reaction. And exergonic reactions release energy. Oh, maybe I need to charge up this tablet pen. They release energy. Now, the material of the glucose and the carbon dioxide is going to be converted into CO2, 6CO2, and 6H2O. Okay? So that's just the physical material that makes up the oxygen and the carbon dioxide, but the energy is going to be stored somewhere else. We're going to take that energy and we're going to use it to combine this coenzyme, ADP, with inorganic phosphate, and that is going to get us ATP, the energy currency of the cell. Now, if everything goes absolutely perfectly, we will end up with 38 ATP at the end of this process. Now, there's a whole bunch of different steps in cellular respiration. So we have glycolysis. We have the transition reaction. My pen's not picking up all the way. We have the Krebs cycle. And then we have oxidative phosphorylation. Oops. Okay. Now, you get ATP from several of these reactions. In glycolysis, you get 2 ATP net. Net because you have to invest 2 ATP at the beginning of glycolysis, but then you get 4 back out. So overall, you end up with 2 more ATP than you started with, so 2 ATP net. The transition reaction doesn't give you any ATP, but it does give you NADH, which then can be used in oxidative phosphorylation. The Krebs cycle gives you 2 ATP. And the Krebs cycle and glycolysis both use a kind of ATP synthesis which we call substrate level phosphorylation. Oxidative phosphorylation, on the other hand, the other kind of phosphorylation that you have in cellular respiration, gives us 34 ATP. So that is by far the biggest payoff, is the last step. Oxidative phosphorylation is the electron transport chain. Usually electrons are abbreviated with lowercase e negative. There we go. Transport. chain. 1 second. Yeah, see if I have a pen that has full charge here. Yeah, that one seems like it may be a little better. Okay, the electron transport chain. So that's the very last step of cellular respiration. So let's go through it by the numbers. We have glucose. Now, what we talked about at the end is that you don't need to necessarily start with glucose. Okay, maybe I just need to reset the computer. It's just lagging. You don't necessarily need to start with glucose. We could also start with amino acids or polypeptides or triglycerides, and those molecules enter this pathway at different points in time. The reason why we start with glucose in our examples is because that gives you the entire picture, the complete picture of cellular respiration, but other materials can be converted to some of these intermediates that we're going to see. Okay. In glycolysis, this is an anaerobic process. So glycolysis doesn't involve oxygen. You break this 6-carbon glucose into 2 3-carbon pyruvates. 2 3-carbon pyruvates. Now, you may sources it'll say pyruvic acid, talking about the same molecule, pyruvate, pyruvic acid. When you see 8, that's just the reciprocal base of that acid. So it's the same kind of molecule, just with or without an extra hydrogen attached to it. So we have these 2 pyruvate molecules. Now, in the process of glycolysis, we get 2 NADH and our 2 net ATP. We have the energy investment phase, which is minus 2 ATP. We lose 2 ATP, but we get the energy payoff phase where we get plus 4 ATP. So this is where we get our 2 net ATP from. Now, NADH, this coenzyme, it's going to be used in the very last step. It's going to be used in oxidative phosphorylation here, but we're not there yet. Glycolysis is an anaerobic process, so you don't need oxygen to make these NADH here. And if there is no oxygen present, well, you've got yourself a problem, because the only way you can make more ATP then is to keep breaking down glucose. Yes, Caitlin, you have a question. Where can I find this recording on Blackboard? Okay, that is a good question, actually. Let me just take a quick break from thing here. I got my Blackboard dashboard here. Okay, I'm going to go to the virtual office hours. No, I've been meaning to make a video about how to do it, but I haven't gotten around to it. So, and then if you see this little sandwich menu here, this little hamburger menu, so don't go into the course room, just click on this menu, and that's where the recordings are found. And then you can see that we have 3 of them already, and this will be recording 4 for this class session. You're welcome. All right, back to action. All right. So, if there is no oxygen present, so if there's no oxygen, and when we say oxygen, we mean O2. Right? If there's no O2 present, then we have a problem, because the only way we can get more ATP is to keep doing glycolysis. I don't care what's trending on Reddit. Is to keep doing glycolysis, But in order to do glycolysis, you actually needed this coenzyme, NAD+, in order to make these two NADH. It's not a convenience, it's a required part of glycolysis. So we need more of this. We need more NAD+. But the only way that traditionally you would get rid of NADH and remake NAD+ is through oxidative phosphorylation. But that requires oxygen. So what's the cell going to do? Get rid of all these extra arrows here. What's the cell going to do? Well, it's going to take the H on the end of this NADH, and it's going to reduce pyruvate. Okay, it's going to reduce pyruvate. So if pyruvate is reduced, reduction, you will get either ethyl alcohol plus carbon dioxide, CO2 is another byproduct of this, or you'll get lactic acid. Our cells make lactic acid, and this is called fermentation, right? This whole process is fermentation. And our cells make lactic acid through fermentation. But if you have like yeast and some other cells, they make ethyl alcohol plus carbon dioxide. In the video, the cellular respiration videos where I'm baking a loaf of bread, right? The reason why that bread rose so much, where you got all those nice bubbles and all that, is because of this byproduct of fermentation, because of all that CO2 that came through. So that's where we got that. The purpose of fermentation is to make more of this coenzyme, to make more NAD+, because otherwise, if you keep doing glycolysis, you're going to run out of it because it's all going to be converted over to NADH, which you can't use for glycolysis. So you have to get it back into its original form. So that's something to remember, is that the purpose of fermentation is to recycle NAD+, to get more NAD+. Okay. Now let's say that we move past glycolysis. Right, so we got 2-pyruvate. Glycolysis happens out in the cytoplasm of the cell. It happens outside of the mitochondrion. The mitochondrion is going to take responsibility for all of the aerobic respiration processes in the cell. So the pyruvate can then move into this mitochondrion. But as soon as it gets in there, we get the transition reaction. Transition reaction, which creates an additional 2 NAD, 2 NADH, I made this canvas too large. That's why it's lagging. And you lose a CO2 in the process. The pyruvate then is converted over to, instead of 2-pyruvate, you get 2-acetyl-CoA. Okay, so we lost a carbon dioxide. We gained 2 more NADH. Our total is 4 NADH now. And we've made 2-acetyl-CoA, which then can continue, the acetyl-CoA is here, which then can continue into the matrix where the Krebs cycle happens. Okay, so the Krebs cycle, these are all processes that will occur if there's oxygen present. The Krebs cycle takes acetyl-CoA and it passes it around right? Officer, can I keep selling sugar for money? And through the process of the Krebs, you don't need to remember all of these intermediates, by the way. I just wanted to make sure I had the right number of steps. In the process of the Krebs cycle, you are going to lose 2 more CO2. And you're going to gain 2 more ATP, again through substrate level phosphorylation, which just means that we're using some enzymes to directly move phosphate groups from one molecule to another. There's no ATP synthase involved. There's no chemiosmosis involved. Substrate level phosphorylation is very straightforward. Just tear a phosphate from one thing, pop it onto another thing. All right, so you get 2 ATP. But you also are going to get 3 NADH per acetyl-CoA, and an FADH2 for every acetyl-CoA. Now, we started with 2 acetyl-CoA, right? Because if we go back to, where are we? If we go back to the transition reaction, we converted our 2-pyruvate to 2-acetyl-CoA. So now they have entered the Krebs cycle in the mitochondrial matrix. Each one, we've lost some CO2, so technically, I guess, if I were to make this totally consistent, you get 4 CO2 coming out, and then instead of this, I'll say that since we have 2 acetyl-CoA, that's going to give us 6 NADH total, and 2 FADH2. Right? So 2 times gets you that. Okay, so that's the Krebs cycle. Oh, Caitlin, do you have another question? Or is that your hand up from earlier and I never put it down? Oh, okay. All right. Let me know, because I get carried away. You guys know that. All right. So what do we have so far here? I'm going to change my color. I got 6 NADH from the Krebs cycle. I got 2 FADH2, and I got 2 ATP. From pyruvate oxidation, I got 2 NADH. And from glycolysis, I got 2 NADH and 2 ATP. So let's get some totals going here. I have 4 ATP. I got 8. How many questions will be on the test? 25, 30 for the cellular respiration test. I don't remember exactly how many there are, but somewhere in that ballpark. The tests are much shorter than they otherwise would have been, but there'll still be a good number of questions. Yep. We got 4 ATP. We have, well, math is not working this early in the morning. There we go. 10 NADH, right? Because we had 2 from glycolysis, we had 2 from the transition reaction, and we got 6 from the Krebs cycle, so that's our 10 NADH. And we also have these 2 FADH2 enzymes. Now, remember the anatomy of a mitochondrion? I did find it, that is a terrible mitochondrion. I did find a way to upload a diagram, so there will be a labeling the mitochondrion portion of that test. So we have the outer membrane. Okay, I'm still learning here, so now I know to make smaller file sizes so it doesn't lag so much. Then we have this folded membrane, which is the inner. The inner membrane here. And inside, deep inside, is the matrix, right? That's the interior of the inner membrane. And the matrix is where the Krebs cycle happened. But the electron transport chain happens on the inner membrane. It happens across the inner membrane. Okay, the electron transport chain there. Now, the electron transport chain, it's kind of like the light-dependent reactions where you have this membrane with all of these different protein complexes and coenzyme, enzyme complexes and coenzymes in it. And then we have ATP synthase. Which is able to make ATP using chemiosmosis. And how does chemiosmosis work? You have to pump a high concentration of hydrogen cations, positive hydrogen ions, to one location so that they want to fall down to an area where they're less concentrated. So NADH is going to donate its electrons to the first pump in this chain. FADH2 adds its electrons onto a slightly later step. It gives them to this complex number 2, which then goes here. The electrons from NADH go from complex 1 to complex 3 to complex 4. And eventually, all of these electrons that they're carrying around, right? We captured energy from sunlight way back in photosynthesis, and we use that sunlight to charge up these high energy electrons. Well, the glucose has been carrying those high energy electrons this whole time, but we finally transferred all of that energy, all those high energy electrons, to these two coenzymes, NADH and FADH2, and they're now transferring those electrons to these protein complexes. Now, these complexes pump hydrogens. Right? So if you have hydrogen ions sitting around, it's going to pump them from one side of the membrane to the other. NADH does complexes 1, 3, and 4. So you get a hydrogen for each one of those. So for every NADH, you get 3 hydrogens pumped into the intermembrane space. For every FADH2, you get 2 hydrogens pumped into the intermembrane space. And there we have 10 of these NADHs, so we're going to get a lot of them. So 3 for every 1, 2, 3, 1, 2, 3, 1, 2, 3. I've entirely lost count, but the point is we're going to be making a lot of positive charge, and we're just constantly breaking down more molecules, making more NADH, and pumping more positive charge into the intermembrane space. Now, that creates a lot of potential energy because these high energy electrons, they want to get away from each other. So they're going to fall back down their electrochemical gradient through ATP synthase, but every time we get one hydrogen moving through, we get one ATP. So ATP synthase takes ADP plus inorganic phosphate and gives us an ATP, in this case through oxidative phosphorylation. Oxidative phosphorylation. Why oxidative phosphorylation? Well, remember that part of the equation of cellular respiration involves O2, right? And these electrons that NADH and FADH2 are adding to the electron transport chain, they have to go somewhere. So they eventually end up on oxygen, and we would get, from these two O2 molecules, we would get two H2O. Because remember, that H on the end is a hydrogen. Those high energy electrons are riding on hydrogens, and reduction reactions in biology usually involve not only the transfer of an electron, but also the transfer of a hydrogen. So you end up making water as a byproduct here. Good phrase to remember, oxygen is the final electron acceptor. Of the electron transport chain. Electron transport chain. Okay? It's the final electron acceptor. All of these protein molecules, these complexes, these enzymes here, they're electron acceptors. They're having electrons added to them and they're accepting them, but they're not the final one. Oxygen is going to be the final electron acceptor. That's where those hydrogens and electrons end up at the end, is on oxygen, and that's why we make water out of this process. Ooh. Okay, so what do we have here? And put it about there. All right. We have glycolysis, which is an anaerobic process. All cells do glycolysis. So anaerobic bacteria, the ones that can't deal with oxygen at all, they still do glycolysis. When you run really, really hard and you run out of oxygen, you still use glycolysis in order to break down sugars to get energy. If there's no oxygen present, we go to fermentation, which produces ethyl alcohol and lactic acid, and its purpose is to regenerate NAD+. If there is oxygen present, we go to the transition reaction, right, which is here where we make pyruvate into acetyl-CoA. Acetyl-CoA then goes to the mitochondrial matrix where it's involved in the Krebs cycle, where we get more NADH and more FADH2 and a little bit more ATP. But overall, the biggest explosion of ATP is going to come from this electron transport chain. So we had 10 NADH and 2 FADH2. You get 3 ATP per NADH. And we had 10 NADH. If you multiply that out, so you'll find units cancel and you end up with 30 ATP. You get 2 ATP per FADH2 times 2 FADH2 gets you another 4 ATP. And what does that add to? 34 ATP. plus the 2 that we got from glycolysis, plus the 2 that we got from the Krebs cycle. So in total, not 34, 38 ATP total. That's the idealized perfect number of ATP that you could get from this process. We never really get that number. Usually it's going to net somewhere around 34 ATP or 32 ATP, something like that. Sometimes this inner membrane is a little bit leaky, so you don't get 100% ATP production out of these hydrogen ions, but 38 is the idealized number. So on the test, I will use 38 ATP as the total number of ATP that you get out of all of cellular respiration. But you're going to want to know where they come from. So know that you get 2 net from glycolysis, you get 2 through substrate level phosphorylation through Krebs cycle, and you get another 34 through oxidative phosphorylation.